Hello everyone, welcome to Cell Lab. Today we have isolation of DNA from blood by phenol chloroform method. At the beginning of the extraction, we pour 2 ml of blood sample into a falcon. Note that only white blood cells have nucleus. Therefore, only white blood cells have DNA. However, we know that the blood sample contains red blood cells, platelets, and plasma. The liquid part of the blood is called plasma and comprises about 55% of the blood volume. And after separation by centrifuge, the yellow-colored liquid, which is plasma, is placed in the upper part of the test tube and includes the following: water, iron, proteins such as albumin, fibrinogen, collagen, and immune factors. It also contains sugar, fat, dissolved gases such as carbon dioxide and oxygen, and also hormones, waste materials, and etc. Red blood cells, also known as erythrocytes, contains no nucleus, just like platelets, and play a role in oxygen transport. However, white blood cells, leukocytes, have a nucleus, and after separation in test tube, they are seen as a white part under the plasma. In the continuation of the test, the red blood cells in the sample should be lysed. For this, a volume of 2 or 3 times the amount of blood should be distilled water added to the sample that causes water to pass through the wall of the red blood cell, and with the increase of turgor of the red blood cell and eventually rupture of it, red blood cell formation occurs. So add water to the falcon to a volume of 8 ml and then shake the falcon for 5 minutes. Now 2 falcons should be placed in the centrifuge and rotated. We centrifuge at 5000 rpm for 5 minutes. After centrifugation, we observe that the white blood cells at the bottom of falcon tube have collected and the red blood cells were lysed, and because they have a lower density, they are seen in the upper part. We slowly drain the supernatant containing red blood cells from the falcon tube by inverting. Now, to ensure the lysis of red blood cells, we repeat the above steps again. That is, we add white blood cells to the sediment, centrifuge at 5000 rpm for 5 minutes, and then drain the supernatant solution. We can repeat this 3 times to be sure. Now it is necessary to dissolve the white blood cells and use a buffer called lysis buffer, which contains EDTA, trice, SDS, which is a detergent that dissolves the cell membrane, and we also need proteinase K, which turns our protein into smaller units before they are denatured. As a tip, we know that red blood cells are easily lysed with distilled water, but to lyse white blood cells and separate the nucleus and DNA, buffer must be used. The role of proteinase K is that this enzyme removes proteins and enzymes to destroy enzymes that damage DNA during DNA purification. In the continuation of the test, the red blood cells in the sample should be lysed. For this, a volume of 2 or 3 times the amount of blood should be distilled water added to the sample that causes water to pass through the wall of the red blood cell, and with the increase of turgor of the red blood cell and eventually rupture of it, red blood cell formation occurs. So add water to the falcon to a volume of 8 ml and then shake the falcon for 5 minutes. Remove the upper phase slowly and pour it into a new microtube, the same amount of chloroform, and add the possible removal of the remaining lipid and shake the mixture for 15 seconds at 12,000 rpm. Centrifuge for 10 minutes and remove the supernatant slowly and pour it into a new microtube. Finally, as much as we have 8% alcohol propanol and 1% sodium acetate, add and invert the sample a few times. DNA is visible, and now we centrifuge the sample at 12,000 rpm for 10 minutes until the pellet reaches the end. Deposit the microtube, then empty the supernatant solution and add 70% ethanol alcohol. We add it to the sample so that the sample is soaked and spin in centrifuge at 14,000 rpm for 5 minutes, and then empty the supernatant and repeat this step again. Note that the DNA is soluble in water, but in the presence of salt and alcohol it is insoluble and is seen as precipitate. Next, to dissolve the DNA plaque, trice buffer, and EDTA. This buffer is called TE, used for long-term storage of DNA. At the end, subscribe for Cell Lab on YouTube.